Most of the research done in cancer in these days is based on a genetical approach. Certainly, genetics has an important paper in the production in the sickness cancer, but there are other mechanisms important to that. The first mechanism that we are going to see in this presentation is the metabolic mechanism, its thermodynamic basis, and the consequences on the information system. The healthy cell represents optimum conditions for life. First, there is an enormous energy flow. This energy flow is caused by glucose that when it's burned produces ATP. ATP is a form of energy that is exported out of the cell via the sodium-potassium pump. Another very important healthy condition is that the membrane resting potential is of minus 70 millivolts. And also another condition not very well identified is that there is an information flow that implies negative feedback. That is information. A healthy cell has a very strong membrane resting potential of minus 70 millivolts, and when it repolarizes, it goes up to plus 30 millivolts. This is a large energy flow. This energy flow implies electric current, implies open communication and information channels with the rest of the body. As you can see in the curve, the energy release is represented by the surface below the repolarization curve and is exported out of the cell as information, as electrical information. It would be impossible to have an organism, to have a tissue, to have a body without this information. On the contrary, a sick cell doesn't have such a strong membrane resting potential. The membrane resting potential goes to minus 10 millivolts. The cell continues burning glucose, but doesn't burn most of the glucose through the Krebs cycle, but through the Warburg cycle. The Warburg cycle is like a fermentation cycle. We will see that later. So, as there is no potential difference and the repolarization curve is much smaller, the sodium-potassium pump doesn't work efficiently, and there is no energy or potassium ions output. The potassium-sodium pump doesn't work properly, and there is no information output or feedback. The cell sort of divorces from the rest of the body, from the rest of the system. A very important issue that the sodium-potassium balance is modified. The cell contains excess sodium. The cell contains excess sodium and is deficient in potassium. So the potassium pump becomes inefficient and is not able to export the energy produced. Whenever the energy produced is not exported, there is a signaling that tells the cell to change from the Krebs cycle to the Warburg cycle. The Warburg cycle, which was discovered in 1930 and gave Warburg the Nobel Prize, is a fermentation process, producing lactic acid in an acid environment. The resting membrane potential goes down to minus 10 millivolts instead of minus 70, and there is no repolarization, no messaging, and no information feedback. Excess sodium and excess glucose is a common factor in 21st century diets. When we talk of a thermodynamic or physicochemical analysis, we are going to talk of open systems far from equilibrium. Open systems means a system that is receiving and expelling matter, is receiving and expelling energy, is receiving and expelling information. First, we have to understand the phenomenon of life. In order to understand the mechanism of cancer, we have to understand how do we define life. The cell is a living entity, and we can define life as the active status of living beings, as specifically distinguished by the capacity to grow, metabolize, respond to stimuli, adapt and reproduce. and probably we should add subject to evolution, comprises a complex system which auto-organizes and is in thermodynamic non-equilibrium, takes energy and matter from the surroundings, excreting them in a degraded form. Cells represent an open thermodynamic system, the most exemplary non-equilibrium thermodynamic system, receiving energy from the surroundings and transforming it in free energy meaning energy able to do work, which is then transformed into life and order. To produce this order, we need large information flows. Once this energy is used, it is rejected as leftovers. Life is considered to exist when each of the following characteristics is observed: order, evolutionary adaptation, response to environment, regulation, energy processing, reproduction, growth and development, information exchange. All these represent non-equilibrium conditions. These are the main characteristics of life. In the 40s, the famous physicist Erwin Schrödinger, who was one of the developers of quantum mechanics, began to study life, and he produces a small and still very famous book which is called What is Life? The implication of this last paragraph, the paragraph of the previous slide, is that life is a phenomenon that needs a different analysis than the one given by classical thermodynamics, but the old thermodynamics, creating order out of disorder and using energy derived from burning sugar. So the burning of sugar should be carried in such a manner that the main product is energy usable for production of work and not heat. In the case of cell, the burning of sugar produces electrical energy, at least in the case of neuron cells, carried outside the cell by the sodium-potassium pump. When working on this presentation, I reviewed various articles and videos. In one of those articles, the famous oncologist defines life, that life consists of a structure accompanied by energy flows and information flows. Information is inserted in the DNA, but it has to flow. Inside the DNA is like information inside the DVD. The information is not available. The cell, on the contrary, sends information and receives information. In order for information to flow, there has to be a message that has to be communicated. A sender, a receiver, and a feedback loop. Non-equilibrium condition life means that we go from the most stable state, which is the existence of carbon dioxide plus water, to the formation of very complex molecules, proteins, ADN, or DNA, in Spanish. On the contrary, classical thermodynamics tells us that the most probable state is the state of maximum entropy. The state of maximum entropy is CO2 plus H2O. There would not exist any complex molecules, so they would burn instantly. Eventually that happens on death. On death, the system becomes unstable. The system goes back to maximum entropy. Contrary to other approaches in cancer, we begin by defining life. This definition is based on the work of many scientists, especially Erwin Schrödinger and Ilya Prigogine. We talked a little bit before about Erwin Schrödinger. Ilya Prigogine was also a Russian physicist living in Belgium, who produces the irreversible thermodynamics, who studied thermodynamics under a different way. This postulate is based on what we call irreversible thermodynamics, and represent the most important one in this presentation. If this postulate doesn't convince you, probably it's not important to see all the other postulates. All the other postulates complement this approach, but the theoretical basis lies in the first postulate. Somehow, information is coded in the DNA. Let us say that the DNA is the cell computer. However, information is flowing in and out, interacting with the DNA. Any important system is composed of organization. In the case of the human body, we can say that we have four complex systems, four levels of organization: cells, tissues, organs, and the whole body. All of them very complex structures communicated via energy or via matter or via information with all of the body, within nearly 20 trillion cells in the body. Very complex structures with very large feedback loops, very enormous and abundant feedback loops. In all those, energy comes from the energy producing mechanism of the cell, the ATP. The ATP is the most important energy and information production mechanism. In the case of animals, mostly for the burning of glucose through the ATP mechanism. The cell, in order to keep its complex structure, needs energy. Part of that ATP energy is used for its own maintenance, and the rest of the ATP, the rest of the energy, is transported as electricity to the cell membrane, using the well-known mechanism of the sodium-potassium pump. The cell, through the sodium-potassium pump, transmits electrical energy to be used by tissues, organs, and the full body, to think, to communicate, to make sports. So essentially you need a good membrane polarity to supply electrical energy to the rest of the complex systems and to transmit information back and forth to the brain. We can see another important mechanism in the electrocardiogram, similar. Insisting on this matter, any organization, whether it is a body, a city, a country, the full world, any organization in order to exist is based on information exchange and feedback loops. Any complex living system needs energy. Part of that energy is spent in maintaining the information structure, and the rest is the necessary energy for messaging or signaling between cells, tissues, organs, and the full body. This is obtained, in the case of living beings, from electrical currents and messenger molecules issuing energy derived from the polarization phenomena on each cell. The most common energy producing mechanism in the cell is the combustion of glucose. Glucose burns in the cell or outside the cell. If it burns outside the cell, it will produce an immense amount of energy. An immense amount of energy that goes to heat. In example, when you are burning wood is a polyglucoside or a polysugar. The temperature that you reach is enormous. It can be higher than 500 to 600 degrees centigrade. In contrast, the burning of sugar in the human cell, which produces the same, CO2 plus H2O plus energy, burns it in a different form, a form that is at low temperature, at the temperature of our body, and it also using a very interesting mechanism. This non-equilibrium mechanism involves very small activation energies to overcome the body temperature, so that the body temperature doesn't rise. If you can see on the right side of this figure, you see the activation energy when burning sugar outside, when burning sugar with fire. You can have an enormous activation curve, an enormous amount of energy that is released as heat. In the case of the burning of sugar in the cell, by the Krebs mechanism, you have the same reactants and the same products, except that this burning is carried by small activation energies. As you can see, many small activation energies, which certainly doesn't produce any such amount of heat. When burning sugar outside the body, when burning it with fire, you have a very large entropy factor. You produce more entropy, produce more equilibrium, and you have a much larger heat and much smaller energy for work. Instead of directly transforming sugar to carbon dioxide and water, the oxidation inside the cell proceeds in a series of steps, the Krebs cycle. There is an intermediate energy carrying molecule, the ATP. The free energy of this reaction, the burning of sugar, in both cases is defined as delta S. The free energy, the energy available for work, is equal to the energy released by the reaction minus the entropy factor. We can see that the higher the temperature, the lower the free energy for work, concluding that in order to inject a large amount of energy to the cell, the burning of sugar has to proceed at the lowest possible temperature. All the free energy produced, all the T delta S factor, which corresponds to non-usable heat production, to entropy, in order that the system receives a large flow of energy and become organized. We need that delta G has a very large value. So the system receives a large flow of usable energy and is able to work under non-equilibrium conditions. Remember that one of the conditions for non-equilibrium is very large energy flows, and thus auto-organize in such a way that we are fulfilling the condition to be in non-equilibrium and self-organize. The sugar burning reaction has to proceed via the ATP-ADP mechanism. That is the only possibility. Whenever we are not using the Krebs cycle but go to another cycle, we are not producing efficient energy and we are not producing large energy flows. A healthy cell is characterized by a strong sodium-potassium pump that moves out 3 sodium ions and moves in 2 potassium ions, producing a difference of potential in the resting cell membrane of minus 70 millivolts, meaning minus 70 millivolts on the healthy cell. When we measure this potential difference in the sick cell, it goes as low as minus 10 millivolts. And this happens when we have excess sodium in the cell and a deficiency of potassium. This pump is driven by energy obtained through ATP in an activated ATP pump. This is a typical cell membrane. In the cell membrane exist several sodium-potassium pumps. We see one of the sodium-potassium pumps. The sodium-potassium pumps takes the energy stored in ATP, makes out of that ADP, and at the same time imports 2 potassium ions and exports 3 sodium ions. If the potential difference in the membrane becomes lower, not enough energy will be transported out of the cell. The cell will become saturated with energy. And one of the conditions for achieving order and non-equilibrium is immense energy flows. If we see the membrane potential difference, say of minus 70 millivolts in nerve cells, in neuron cells, going through a width of 3 nanometers, we have a potential field of nearly 200,000 volts per meter, an immense voltage or energy difference. Just for comparison, look at the voltage carried by a whole voltage line in the street in the order of 130,000 volts. So you have an enormous potential difference for firing the neurons, for firing information, for firing electrical currents. The healthy cell in non-equilibrium dynamics in life adjusts the various physicochemical factors, oxygen, ATPase, glucose, sodium, etc. This thermodynamic system is very delicate, and to maintain this steady state implies balancing multiple factors. Agents known to be carcinogenic decrease the concentration of potassium and increase the concentration of sodium. Anticarcinogenic agents have the opposite effect. Carcinogenic agents include obviously sodium, cadmium, fat, cholesterol, calories, and alcohol. On aging, potassium leaves the cell, sodium enters, and the rate of cancer increases. Of course, we have to consider that if there is not excess sodium, this mechanism for cancer becomes less possible. When having weaker membrane cell potentials in the order of minus 10 millivolts, the energy produced goes mostly to heat entropy, and the voltage field that was 200,000 volts per meter diminishes accordingly to a degree which is not enough to maintain non-equilibrium conditions. When the cell becomes sick, there is a third mechanism for burning sugar, the mechanism that is used by cancer cells, the Warburg effect. the fermentation of sugar to produce lactic acid and proteins needed for the cancer cell to grow. Glucose plus oxygen would give lactic acid plus CO2, and at the same time glucose plus oxygen gives protein plus CO2. If the balance of sodium and potassium does not work properly, then the mechanism for using sugar changes and goes to the fermentation route. Remember, like the fermentation of yeast in a common fermentation reactor. Instead of turning sugar to carbon dioxide and energy, with less oxygen available, it produces lactic acid and proteins, consequently producing less energy, and so having a smaller energy flux to preserve non-equilibrium conditions and to produce electric currents and signaling. A similar mechanism, as I mentioned, as that used to produce yeast. The mechanism used by old cells. Insisting, aggressive carcinomas, in all aggressive carcinomas, is common that they ferment glucose to lactate, even in the presence of oxygen. This particular mechanism, named aerobic glycolysis, the glycolytic phenotype or Warburg's effect, was discovered by the Nobel laureate Otto Warburg in the 1930s. Since this time, controversial discussions about the relevance of the fermentation of glucose by tumor took place, instead of the traceable fermentation. However, a majority of cancer researchers considered the Warburg effect a non-causative epiphenomenon. They say that it exists, but it's not a cause of cancer, it's a consequence of cancer. Recent research demonstrated that several common oncogenic events favor the expression of the glycolytic phenotype. This phenomenon is an inside the cell phenomenon, doesn't need a large membrane potential difference. Under this mechanism, no energy in excess is produced, and so no energy needs to be exported via the sodium-potassium pump. The protein produced is used to abnormally grow the cell. Obviously, the cell becomes independent. It doesn't have any more electricity or information exported to other cells. It becomes an independent cell. It becomes to behave as an independent cell from the rest of the body. The combination of these factors, CO2 poorly transported out of the cell, thus creating acid conditions. You have CO2 plus water, you will create an acid condition both in the lactic acid and in carbonic acid. Water poorly transported out of the cell, thus creating swelling. Recent research and therapies on promoting the death of cancer cells, depriving them of sugar, are being successful. In cancer cells, glucose starvation, when you get into a diet of low glucose, activates a metabolic and signaling feedback loop, leading to cell death. Glucose starvation induces generation of reactive oxygen species, thereby inhibiting phosphatases and activating tyrosine kinases, which in turn generate additional reactive oxygen species. This glucose starvation induces positive feedback loop, amplifies once again the ROS levels until cells undergo ROS-mediated cell death. We spoke about this in the introduction, and we are going to get into a little bit more detail. Under healthy metabolic conditions, energy and matter are transported to the intercellular membrane with a characteristic resting transmembrane potential of -70 millivolts in the example of neuron cells. We might also say that the various information channels work efficiently. When the cell depolarizes and then repolarizes, it fires an electrical ionic current which communicates with the rest of the body. The cell is somehow connected to trillions of cells. The brain has to coordinate them all, and for coordinating them all, you need a signaling phenomenon. So the healthy cell has a signaling phenomenon. The only way to coordinate an organism is through information flows, through signaling. The cancer cell has a much lower membrane resting potential, as it does not depolarize or repolarize. Consequently, it has no signaling and does not coordinate with the rest of the body. Behaves as an independent entity. As we saw in the introduction, we are seeing again the repolarization curve for a typical neuron cell. Remember that the membrane resting potential of this cell was minus 70 millivolts. And then there is signaling. If there is not such a large potential difference of minus 70 millivolts and the potential difference was to minus 10 millivolts, then there will be no information going out of the cell. There will be no signaling. The cell, the cancer cell, the sick cell becomes independent, has a communication problem, is out of control, out of the mechanism of control carried out by the brain. There are no feedback loops. And given its conservation throughout evolution, the action potential seems to confer evolutionary advantages. Remember that the first independent cells did not have a mitochondria, did not have the possibility to communicate, and so did not have the possibility to organize themselves in tissues, organs, etc. One function of the action potential is rapid, long-range signaling within the organism. The conduction velocity of this signaling can exceed 110 meters per second, which is 1/3 the speed of sound. Other conditions causing cancer can have the same influence: pollution, smoking, unhealthy feeding, stress. The cell stops working with an inefficient metabolism and goes to an inefficient metabolism, and instead of producing energy in non-equilibrium and thus life and order, turns to equilibrium mechanisms like the Warburg effect, producing disorder and death. As an example, with the neuron cell, when it depolarizes and its transmembrane potential in the sick cell goes down to minus 10 millivolts, due to strict thermodynamic reasons, a weak membrane will have a lower surface energy, will be less able to sustain swelling, will be weaker, and will try to reproduce faster. Remember that the cellular membrane is not a defined line, but a zone of important potential gradient. Once the membrane weakens, the cell divides faster and easier. Also, when in the fermentation bowl, there is abundant protein produced for growth. Cancer cells are hot cells. The cell, through an inefficient burning of sugar, produces more heat. That heat is not exported, and cancer cells become hot cells. Remember that in the common cell, mitochondria performs the energy producing conditions, and mitochondria has its own DNA. It's a much smaller DNA, perhaps comprising 10 genes or similar. So when the cell changes from burning to fermenting sugar, it somehow needs the permit of the cell. The cell has almost no capacity of sending or receiving information, so easily an isolated cell sets on some switches to produce a sick cell. DNA, observing in both cases negative conditions, informs the cell the need to have a different behavior. There are no feedback circuits to correct the erroneous behaviors or changes in the DNA. The well-known phenomenon of angiogenesis. The cancerous cells, through messengers, convinces the neighboring cells to produce blood-feeding channels, angiogenesis, that permits the feeding, the access of glucose to the sick cells. And long-distance messengers convince other cells, metastases, to duplicate this phenomenon. But if one cell is working with a weak metabolism, all the cells in the body to some extent will also have a weak metabolism. Will also show the same symptoms: high sodium, low potassium, high glucose. You have an inefficient metabolism in one part of the body. Probably, let us say that this is due to a high sodium concentration. Then you will have the tendency of the other parts of the body to also have a high sodium concentration. The sick cell becomes to behave independently. It becomes somehow independent of the several organs, and the loss of cell adhesion and apical cell polarity yields the mobility of cells, trying to escape. The epithelial to mesenchymal transition, EMT, is a crucial process in tumor progression, providing tumor cells with the ability to escape from the primary tumor, to migrate to distant regions and to invade tissues. EMT requires a loss of cell-to-cell adhesion and apical basal polarity, as well as the acquisition of a fibroblastoid motile phenotype, so we can define the cancer cell and the cancer tumor as disorganized from other body functions. A modern therapeutic approach should be oriented toward constructive mechanisms, towards recovering a good metabolic conditions, toward recovering polarizations, towards recovering information flows, and obviously to recover autopoiesis and self-organization. What about destructive methods? If you consider the previous postulates to be true, destructive methods, which kill cancerous cells, will eventually produce more sick cells. But if some cell survives the destructive method, their dynamics and feedback will be towards multiplication. The basic metabolic fact will not disappear. On the contrary, the chemotherapy, irradiation, etc., will produce a weaker metabolism. And obviously all the other metabolic reasons for producing cancer cells will continue to exist with much weaker cells. Weak because of defective metabolism, weak because they are damaged by chemotherapy. Chemotherapy is a method that has no relation to all these analyses. In example, when experimenting with new chemotherapy agents, the usual method is to put a sick cancer cell in petri dishes and find out if a certain chemotherapy agent kills them, ignoring the fact that part of the problem with cancer cells is that they exist inside a complex system, the human body, with a very complicated information structure, with no similarity even to any animal structure. One of the first chemotherapeutic agents was discovered during World War I, when they saw that when giving mustard gas to populations, cancer disappeared, or part of cancer disappeared. This is like trying to fix traffic by dismantling some cars. In contradiction to this century philosophy, the Cartesian approach started with Descartes, obviamente, which studies one complete system, dividing it in its parts. If you understand the behavior of the parts, you understand the behavior of the system. This was a big step in philosophy, a big step in scientific reasoning, but yielded many limitations, those limitations that have been disappearing during the 20th century via modern analysis, which is based on complex system theory. The complete system is much more than the addition of the parts. It implies emergent properties that cannot be explained by the parts. More work should be done on repolarization therapy. The diet to attack cancer should be based on repolarizing, high potassium, low sodium, and the control of glucose entrance, together with the use of insulin, which is a well-known repolarizing agent, or to find other new repolarizing methods. Note in magnetic fields. Magnetic fields behave different with cancer cells, so much that PET and NMR, nuclear magnetic resonance, is a method of diagnosis which identifies glucose. MRI, magnetic resonance imaging, is a method where cancer cells, under the influence of a very strong magnetic field, make cancer easy to distinguish. But certainly this magnetic field should be high, so they can align the spin of hydrogen atoms in the various carbohydrates, fats, etc. And under this excitation, the cell in an excited state has the ability to perform changes in its metabolism, has enough energy to jump the energy barrier and repolarize again. So the energy is supplied by the magnetic vibrating field. Magnetic treatments commonly offered do not use strong magnetic fields. The magnetic fields needed are those that produce effects on the hydrogen atom spin, so as to give the cell enough energy to repolarize, obviously with the proper balance of nutrients and minerals, low sodium, high potassium. Certainly magnetic resonance methods can give the necessary activation energy to repolarize the cell under the above mentioned conditions. When we spoke of the Cartesian method to analyze cancer and to analyze several scientific phenomena, we see that modern medicine has to go more to the basic facts of metabolic medicine, obviously considering the genetic problems. Certainly, probably sodium and potassium are the main factors, but there should be other factors that will appear. But the theoretical basis will be the same: optimize metabolism and polarity. Once again, if we define life as a combination of structure, information flows, and energy flows, then we have to analyze life phenomena in the interaction of these three factors. Any analysis that is not performed as a complex system analysis is bound to fail. If you analyze only structural factors like genetics, then you are not contemplating the full picture. Resuming, the characteristics of cancer cells. Cancer cells have defective metabolism. They turn their metabolism to fermentation, and although keeping an energy production of 2 ATP instead of more than 30 ATP, they are not subject to high energy flows. They have high sodium concentrations, low potassium concentrations, they are hot, they have a very low resting membrane potential, they do not repolarize, do not send signals, and they divorce from the usual body information channels. Who is helping cancer cells? Most industrial foods contain large amount of sugars. In example, one can of coca drinks contains about 50 grams of sugar. Incredible. You don't see it because sugar is able to be dissolved in water up to 80%. And the daily consumption of sugar in drinks in the US and Mexico, which is our case, is above half a liter per day, only of direct sugar. Then you have other polysaccharides as corn, wheat, bread, etc. Sodium comes mostly from food preservatives. The old technology to produce food was based on adding sodium chloride, and today the preservatives are also sodium based. They could be easily substituted by potassium preservatives like potassium glutamate. Certainly, the large industrial corporations have the technology to produce safe drinks, safe everything. 50 grams in a can of carbonated coca drinks. The very well-known physicist Fritz Capra, in his book, The System's View of Life, he says, "The research relating to the genesis culminated in the elucidation of the physical structure of DNA, the genetic component of chromosomes. This triumph of molecular biology led biologists to believe that all biological functions can be explained in terms of molecular structures and mechanisms. This new perspective, genetics, brought a significant shift in biological research. Was the most important finding, the genetic causes of cancer, but a shift that may well turn out to be the last step in the reductionist approach, the Cartesian approach to the phenomenon of life, leading to its greatest triumph, the work that is being done today in genetics, and at the same time to its demise, not considering the other factors like metabolism, thermodynamics, information." Other non-very scientific therapies are successful to a certain extent. Probably those cures are based on vegetarian diets, and so they do not contain sodium and they contain high potassium. Certainly some deprive you of any glucose-containing compound, intending to starve the cancer cells. Others offer magnetic methods, which if accompanied by low sodium could yield good results, but the basis has to be designed on the metabolic approach. A note on the very distant past. Eons of years ago, some bacteria introduced themselves into other elementary cells and producing mitochondria. This is the case of mitochondria. In antiquity, mitochondria, with its own DNA, began to supply the cell with a more efficient energy-producing method. There was oxygen available, and the mitochondria became the energy-producing part of the cell, thus allowing the production of more energy to be exported for information flows. This was a big evolutionary step because it permitted to integrate larger organisms, larger complex systems, and thus being able to produce multicellular organisms, dead tissues, organs, etc., whose main feature is structure, developed through information. There was enough energy to move information flows and produce complex structures. When the cell becomes cancerous, it doesn't use the Krebs cycle anymore, so it does not have enough energy for efficient information flows, and so becomes independent, does not conform to the structural and information rules, loses an organic and organismic autopoiesis. I am a chemist and a chemical engineer with a PhD in MIT and experience in various fields of chemistry, nuclear, etc. But my father, Dr. Demetrios Odi Payares, this work sort of continues the previous work done by him. In the 1940s, he postulated for the first time the sodium diet in heart sickness. It was the first time that sodium was taken out of the diet. Then he developed the scientific method for electrocardiography. Before him, electrocardiography was like artistic, like out of experience. He developed the scientific method of electrocardiography. Still, his book, The New Basis of Electrocardiography, is used in many schools of medicine. He began experimenting in the 80s in high potassium, low sodium diet in cancer patients with great success, and used what is called the GIK solution, glucose, insulin, potassium. When he found the success of this diet, just on a clinical basis, together with the very famous Henri Lavoit, developed the physicochemical basis of the heart cell. Later, he was in close contact with Scu, the discoverer and Nobel Prize winner of the sodium-potassium pump. Probably he missed the role of sugar in the diet, the information paths and the systems approach. He was also within the Cartesian analysis. His last book, where he speaks on cancer, is called What I Have Found on the Cancer Cell, and even it's only in Spanish, and even if it is a very scientific book, it also implies many personal and philosophical analysis and poetry, etc. It's a difficult book to grasp. It's a book produced by humanists together with by a physician. In his book, Another Policy Actor, you can see several examples of this therapy in cancer, several clinical facts important. During his life, he published more than 500 scientific articles. This is in the 40s to the 90s or to the 2000, mostly in the sector of cardiology, and 20 books in medicine, also in poetry and philosophy. Thank you.